All right, good evening, everyone. Welcome back to our October Café Scientifique. Yes, well, no, and we're doing pretty well now. We should be regular from now on, 2 talks a month. So we'll tell you at the end of this talk, what next week's talk is. But let me first of all just welcome you all and welcome those people who are on the live stream. Sorry, I'm a little bit blinded by the lights. That's why you're probably blinded by the glare off my forehead. So I'm probably doing everyone a sort of a retinal damage. My name is John Willis, and I'll be your host for this evening in Café Scientifique. Our speaker this evening is Professor Karen Helbing, and she is a professor in microbiology and biochemistry at UVic, and she's going to give us a wonderful talk this evening. But before I introduce her and give you the title, let me just tell you a lovely email I got this week, right? And this was an email from Marjorie Wilder, who many of you might remember. She has, for many years, she organized Café Scientifique, and she actually was one of the people who helped start Café Scientifique at UVic back in 2008, she told me. It was May 2008. And, what I didn't realize, I knew that Karen had given a couple of talks for us before. Karen is speaker 0. She is the very first speaker we ever had at Café Scientifique. Apparently Marjorie had to go around town. There was no email list back then. There was no website. So she went around plastering posters up wherever she could get them, trying to rustle up interest. And, she had 30 people for the first talk, right? Which was a good kickoff, right? The second talk had 5. But a local reporter came and wrote up a piece on the evening's talk, and then from then on we've been sold out all the way through. So anyway, thanks for actually. Who was here at the beginning? Who remembers that? We've got a couple. That gentleman there, I think that's about it, and that lady there in the front. I was just curious. I've only been involved in Café Sci since about 2016. Which feels like an eternity ago. I guess anything pre-COVID feels like an eternity ago, but. Yeah, that's the extent of my institutional knowledge. But anyway, I just thought I'd pass that on because I thought it was kind of cute, her experiences and this kind of oral history I find really fascinating. So, with no further ado, please join me in welcoming Karen Helbing, who is here to talk to you this evening about tracking invisible species in our environment. Thank you very much, everyone.
The ritual unrobing here. Ooh, these are very bright lights. Well, it's wonderful to see you all like in actual person, and also those that are virtually attending. Welcome. You're the fearless ones and enjoying the food very much so. Well, it's my pleasure to be here today to talk with you about some of the really cool stuff that we're doing in the lab. A little bit different topic than my first Café Scientifique. Today we're going to talk about, tracking the invisible species. Well, what does that mean, tracking the invisible species? Well, what I'm going to talk to you about today is a bit of a launch of as John so aptly put it, DNA month. And I'm going to talk to you about DNA that is actually released into our environment that actually allows us to do something like CSI for wildlife, to give us an idea about what species have recently been in an area without actually having to see them. So why is that important? Why is that interesting? Well, first of all, our understanding about things like the impact of climate change, the impact of natural resource development, the impact of pollution, all of those things, we turn towards fish and wildlife, for example, in order to detect if there are changes and to monitor biodiversity as an indicator for ecosystem health. Well, how do we know when a species is there or not? Like an at-risk species, for example, or heaven forbid, an invasive species that's just about to take hold. Well, the earlier we can detect changes or impacts, the greater the chance is that we can actually do something about it. How do we see these species? Well, a lot of the species that we're interested in that fall under those categories are really hard to find. So imagine looking for, let's say, a tailed frog. Now, these are really neat frogs. They live in BC, and they live in very rushing waters right beside rapidly flowing streams. They're very cute. For those of you who like amphibians, they're super cute. They have a little, what they call a tail, but it's actually a modified sex organ. And these little guys and girls are happily in those streams, but they're very difficult to actually observe. First of all, they live in remote locations, and second of all, you have to pick the right time of year in order to actually observe them. And then you have to actually know where to look, and then you have to be lucky enough to actually spot one. So you imagine going into the Rocky Mountains and trying to find something that's about this big. Okay, well, it's challenging, to say the least. So colleagues of mine have been like monitoring tailed frogs for quite many years, and there was a fairly recent study that took 4 years, and they looked at, over the 4-year period, they looked at over 200 sites where they had their best guess as to where these wonderful little frogs lived. And during that time, they had about a 7% or so success rate in actually observing the little frogs. Well, what did they have to do? They had to hike around to these remote locations, and then they had to, look under various rocks and other paraphernalia in order to try and find them. Now, what if they could just take a scoop of water from the stream and measure to see if the DNA from tailed frogs happened to be in that water? Well, that would be a lot easier, wouldn't it? So that's actually what they then did. And they then filtered that water, brought the filters back to my lab, and then what we did was actually tested for the presence of DNA from that tailed frog. And in doing so, what took 4 years and a very low success rate suddenly took 5 days with about a 76% success rate and a lot less effort. Now, I've got lots of other really cool stories about how measuring DNA in water instead of trying to find these animals visually actually is a game changer. Another one is some of our indigenous colleagues that live in Alberta area, they were interested in knowing where the overwintering sites of Arctic grayling was. Arctic grayling is a very important fish for indigenous peoples, and also, I'm told, very tasty as well. And knowing the overwintering sites is important in order to protect the species. Now, how easy is it to actually observe a fish under a few feet of snow? Well, not that easy. So what we decided to do was a semi-crazy thing to do, which actually turned out to work. And that was to actually make a hole in the river ice and grab a scoop of water from the streams that, through extensive indigenous ecological knowledge, actually suggested that, yeah, maybe the Arctic grayling did actually overwinter in those areas. And we measured the DNA in that water. And yes, in fact, we were able to show that that in fact we could measure DNA in that water that indicated that the Arctic grayling were hanging out there in the wintertime. So that's pretty cool. So using DNA that's left behind is essentially revolutionizing how we monitor fish and wildlife species. In order to do that, what we do is we utilize a technique that's called quantitative real-time PCR, polymerase chain reaction, or QPCR for short, because that's a lot of words. And it's the same technology that is being used to test for COVID. And this technology is one that actually is highly accessible to many people. And one of the things that we're doing in terms of creating these tests for a whole slew of different wildlife species and fish is making the tests robust so that anyone who is interested in being able to test for these animals are able to do so. And that's particularly important for our Indigenous friends. And just recently, we were awarded a project from the Genome Canada and Genome BC and Genome Quebec to do a pan-Canadian project to actually augment the necessary DNA resources that one needs in order to develop these robust tests for a whole slew of wildlife species. This project, which is starting like right about now, it's a 4-year project, and it's valued at about $12 million or so, and involves collaborators from all over Canada, particularly in BC and in Quebec, and 4 Indigenous partners that are going to be using these new techniques to measure DNA in water. And I should say that this DNA measuring technique is called environmental DNA, or EDNA. Or for some people they call it EDNA, but that's somebody completely different. So with eDNA and this iTrack DNA project that we're just about to start, we're super excited about doing that, because what we're going to be doing is creating all these new tests to actually look at species that are at risk and ones that are invasive or culturally or economically important for remote communities. So I have one more story that I want to tell you about how eDNA techniques have actually changed how we actually look at who's living where. So this is a story about the sharp-tailed snake. Does anybody know about the sharp-tailed snake? Yep. Nope. They are also super cute. They're really small. They're about the size of a quarter, kind of coiled up. And they happen to be an endangered species. That's probably why you haven't heard about it. And they live in places like Salt Spring Island. And as a matter of fact, there's a population of sharp-tailed snakes that live on the side of Mount Tuam. Now, if any of you have hiked along on Mount Tuam, it's pretty steep. Pretty steep. And these cute little snakes, they actually are what they call fossorial snakes. So that means that they like to burrow in the ground, and as a consequence, they're really hard to spot. So biologists have been tracking them and determining like where they live and stuff like that on Salt Spring Island by taking asphalt shingles, like the kind that you have on your roof, and placing those in strategic locations on the side of Mount Tuam. And then they stick a rock on it, and then every once in a while, like on a sunny, warm day, they'll actually walk, or like sort of walk sideways because it's very steep, and go up and down the side of Mount Tuam, and then they literally go to the asphalt shingle and then look underneath and see if they spot a snake at that point in time. Now, as you can imagine, that's not terribly successful in terms of like getting the right time to see when the snake is there. And over a 2-year period, after 6,000 visits doing this, with lots of different, over 100 different, what they call artificial cover objects, AKA asphalt shingles, looking for these cute little snakes underneath. The success rate after 6,000 tries was 0.8%. It's pretty amazing that they could find any. But this is the standard methodology, so the conventional way of looking for these wonderful snakes. So what we did was we developed a technique for taking a cotton finger cot, you can buy in the pharmacy, put the little cotton finger cot on your finger, take a little bit of rubbing alcohol, and then swab the underside of the artificial cover object. And the idea there is that, well, if there's DNA there, we can capture that with our little finger cot. So what we then did was we brought those finger cots back to the lab and then used our QPCR technique to see if we could detect sharp-tailed snake there. And lo and behold, yes, we could. That was pretty amazing. And I was super, super surprised. I did not know this was I was not 100% sure this was going to work. But I'm totally sold on it now. Success rate. Anybody want to guess what the success rate was for? 95%. Oh, thanks. I wish. 95%? Not quite. 60%? Not quite. You're very optimistic. I like you guys. Thank you. There is so much we can't see. It's so true. So the success rate that we had with a lot less effort, so remember 6,000 times doing this? So let's try 82 times, and the success rate was 10%. So that was pretty darn amazing. Yeah. So, cool. There's lots of different applications I'm sure you can imagine for detecting invasive species. Can you imagine the invasive zebra mussel? We don't want that coming to our coast. But there's things happening there. The trick is you want to catch it as early as possible. So this kind of technology can do that. and hopefully give us a kicking chance at actually being able to mitigate an invasion. So we're working with scientists in developing those types of tools as well. So, yeah, so how much time do I have there? Oh, I'm doing good. All right, fantastic. All right, so how does this technique work? Well, so what we're doing is we're measuring DNA. So when I say the word DNA, what do you think of? Double helix? Anything else? CRISPR. Ooh, that's fancy. Pardon me? Screening. Yeah, screening to capture like genetic screening, that kind of thing. So what a physical embodiment of DNA is, we think of that beautiful double helix, right? It's like the classic paper from Watson and Crick. Do you know that it was only 500 words? It was short. It was like the best, most impactful essay ever. And it had a very simple diagram. It was a simple double helix. That was it. That was the paper. Well, what does DNA really look like in the lab? Well, if we isolate DNA from, say, tissue, or like a strawberry or something like that, it looks like snot. So much for the elegant part of it. But in actual fact, that's a lot of DNA all in one go. But when we're sampling DNA from water samples, we can't see that. It just looks like water. So it's very, very dilute, very, very low concentrations. So the method, this QPCR method that we're using, is a very, very, very powerful method that involves taking that DNA and then making many, many copies of it in a very specific kind of chemical reaction. And it's kind of like taking Velcro and taking little pieces of DNA as like specific Velcro that gloms onto the DNA that we care about. And then once that Velcro kind of gloms onto it, that's sort of the specificity of the QPCR reaction. Then what it does is it makes lots and lots of copies of it. So many that we can actually detect it in our little tiny tube. Literally a drop of water. Okay, so when we use environmental DNA, there are some challenges that we face. It sounds so amazing. It's revolutionary, transformative, totally. It really is. But so it measures DNA. Well, what happens if that DNA has been there for a long time? Like ancient DNA. How many of you have watched Jurassic Park? Yeah. That ancient DNA, it's preserved in the amber, Well, it turns out that environmental DNA can be preserved too in like sediment, so sediment cores, for a very long time. So there are ways that we can actually get at that ancient DNA to give us a historical picture of maybe what species have been around for, a long time ago that may not exist in a particular area. So we're doing a project right now. We're starting a project with Alberta Environment and Parks and Environment Canada and some Indigenous First Nations groups that we're looking at trying to find species that used to be in lakes that are impacted by oil sands. So from a traditional perspective, these species were supposed to have been there. You know, there's oral history that these species were in those lakes, and they want to use this EDNA technology to look at sediment cores that have been the sediment cores have been dated, carbon dated, and then we're going to be actually testing to see if we can detect those species that they can't find in the lake anymore. So that's pretty cool. Looking forward to doing that experiment. Now, I've been talking a lot about, well, all about DNA. DNA is very stable in the environment. And that's why, you know, like in sediment cores and stuff, that's why we can do these types of experiments. But like in water and that, they don't last quite as long. There's like bacteria and stuff that like to kind of munch out on the DNA and scavenge it for themselves. But there's another challenge that we have to face with eDNA, and that's knowing whether, well, one thing that environmental DNA or eDNA can't do is it can't tell us whether the organism that sloughed off that DNA from their cells into the water, let's say, whether that animal is still alive or not. So how do we deal with that kind of a conundrum? Well, one is we just sort of shrug our shoulders and walk away. But no, we have another trick up our sleeve. And that trick is a related molecule called RNA. So DNA, deoxyribonucleic acid, the double helix. RNA is ribonucleic acid. Does anybody know what the chemical difference is? It's a quiz. Yes. Very close, yes. The difference between the two are simply a hydroxyl group, OH, oxygen, hydrogen. That's it. That's the only difference between the two, and it's stuck in a specific spot on the molecule. The difference, though, is that what RNA is used for in the cell is it's like the messenger. So if DNA is the blueprint and kind of the boss of the cell, it has all the instructions that the cell possibly could use in order to do its activities. Then the RNA reads specific locations on the blueprint and then translates that information, brings that information from the nucleus in the cell out into the cytoplasm outside to get the cell to do the work. So it's seen as a messenger, or maybe middle management, I don't know. And the RNA, its half-life, it's probably very similar to middle management, I guess, now that I think about it, because RNA half-life isn't very long. I'll have to think about that. Anyways, the half-life of RNA is pretty short. So that actually gives us a little bit of an in on how we could possibly detect animals that have been recently in an environment, like really recently, because that RNA can't hang around for very long. So this is called eRNA, and well, we're just in the infancy in terms of how to harness that power. It's a little trickier, as you can imagine, if something's very short-lived, to try and detect that. But a lot of us are working really hard on trying to figure that one out. But then, what we are able to do is to say, now this fish was there very recently because I've detected eRNA there. But not only that, it was alive because the RNA wouldn't survive very long if the cell was dead or the organism was dead. Yeah, so then on top of all of that, what we're also doing is using, so we're trying to determine if organisms were alive or dead, whether they're there or not, trying to figure out where they are, under what conditions. And all of this sounds really amazing, but one of the things that we're also putting a lot of effort into is actually making sure that it's done well. So you can imagine that, something that sounds really amazing, lots of people want to jump on that bandwagon and, make it like, oh, it sounds really great, I want to do this. But they don't do it very well. So one of the key things that also in this iTrackDNA project that we're doing is we're also developing standards and training modules and so on to make sure that people that are collecting samples and interpreting the results are actually doing so in a good way. So, yeah. All right, so with environmental DNA, what we also are able to do is get an idea about diversity. So not only can we say, oh, well, who was there? But we can get an idea about what kind of community was there as well. Now this takes a slightly different kind of approach. So I talked about QPCR. That methodology is targeted. So we say, oh, I want to know about sharp-tailed snake. Oh, I want to know about salmon. Oh, I want to know about Arctic grayling, etc. With another form of environmental DNA analysis, what we can do is actually ask about, well, okay, I want to know all the DNA that's in there. I want to know all the fishes, and I want to know it now. And what we can do is actually harness the power of another technique that is a mainstay of sort of the genomics revolution. And that is the use of next-generation sequencing. So this next-generation sequencing is the technology that was like massively developed in the Human Genome Project. Do you remember that? Like when the big announcement of sequencing the human genome came out? That was pretty amazing. Well, they managed to sequence the human genome way faster than originally anticipated. They were actually early in finishing that project. Can you imagine? Like something actually finishing early? Forget it. So the reason why that happened was there were massive developments in DNA sequencing technology that happened to meet the demands of sequencing that human genome. The 3 billion base pairs that comprise the human genome. Well, the outfall of that is that we can now use the same technology, tweaked a little bit, a little bit better. It was a while ago, when this happened. But to be able to actually identify, again, take that DNA from that scoop of water and sequence all of it. See what's in there. Now, it's a little bit trickier than the way I'm saying it, but the bottom line is that that's the way we can discover whether there's new species there or not. We might not know what they are quite yet, but we can certainly start kind of putting the pieces together, and with more DNA sequence resources that we're developing all the time, we have databases where we store a lot of information about DNA sequences from what we call voucher specimens. So specimens where somebody, I voucher that this is really a frog of this species, right? Voucher specimen. And then that information is put into a database, and then we can access that. So you can imagine, you go out to, let's say Salish Sea, or the other side of the island kind of thing. Take some water samples and sequence the DNA in there. Get those AGTCs, in order and stuff. And then identify, compare to the databases and say, oh yeah, okay, there's, there's orca, there's salmon, there's, seven-gilled shark, there's a whole bunch of things that they can identify. And then there's going to be a whole bunch of other stuff that they can't. But they can get an idea about relative abundance, maybe, if a sequence comes up a lot of times, that means that there's probably more of whatever that is in there. And maybe eventually we'll be able to actually get a better idea and put the pieces together. And so another activity that my lab is undertaking is actually working with scientists on trying to look at marine environments and to try and get a better idea of what we can't see in the deep ocean. Very exciting there too. So, being able to look at things like sharks and sablefish and other, deeper sea organisms. So, yeah, it's pretty exciting what we're embarking on in this journey of ours in trying to really see those unseen organisms by looking at what's behind, what did they leave behind? They sloughed off their cells and left behind DNA, sort of like a trail of breadcrumbs. Perhaps a little smaller though than breadcrumbs, but still a trail. So I think I'll finish there, and I'm more than happy to entertain any questions that you may have about this exciting thing called eDNA. Yes?
Well, the thing that really interests me the most is I've never given any thought to DNA and water. And I've heard about, in forensic science, touch DNA and all that. The thing that I find so fascinating about this, because water as a medium is always flowing. So you do a sample of water and you find all this DNA in there. And you think that DNA, that creature was there. But in a way, that creature could have been passing through some time ago. And it's only if you find the RNA that you know the creature was there recently. So my question is, how do you sort that out except with RNA? Because you would know that they were there, but it could have flowed in with the tide. The tide goes in, the tide goes out. But it's so fascinating. I never thought that you could do this DNA in water. That's very fascinating. So my question is, except from the RNA, that's the only way you can actually tell the timing of when whatever DNA that is was there recently. Is that true?
So the question was except for the eRNA,
Yes.
The eRNA, how can we deal with the timing of whether an organism was recently there or not? Can we use eDNA for that too? Under what conditions? Is that a fair way to?
More or less.
Okay, cool. Yeah, and you know what? It's a challenging question. So we've got a lot of people working on that concept right now, like how to put the pieces together. The short answer is it's going to depend, I think. It'll depend on the context in which the measurements are made. It'll depend on the environment that is being measured. So you can imagine, taking a water sample is going to be a little bit different in terms of how one interprets the results in a rushing stream versus a more stagnant marsh.
Yeah, tidal pool.
Tidal pools, exactly. Tidal pools, lakes, and so on. So context is going to be very important in terms of making some of those judgments, as well as knowledge about the hydrology and sort of the physical chemical properties of sort of where the samples are being taken.
Yes.
Yeah. Another thing we think is going to have an impact is types of bacteria too, and we're checking that out.
Into what in bacteria?
Types of bacteria. Yeah. Yeah.
There's
Yes.
a gentleman there.
It seems as though sample collection must be one of the major challenges with this. You mentioned, using the cotlets for shark-billed snakes and so on. And I was thinking about invasive species like giant hornets and so on, and it would be neat if you could do that, but it must be incredibly difficult to get your hands on the DNA, I would imagine, from that sort of critter.
Sure. Yeah, so the sample collection, I'm just going to paraphrase, hopefully properly, paraphrase your question. So taking samples and measuring DNA can be very challenging. For example, your example was wasps or the hornets, and trying to measure DNA from them and somewhere. It can be very challenging. So some species will slough off DNA more readily than others. You can imagine like a frog or a fish might more readily slough off DNA than say a rattlesnake or perhaps a hornet or a wasp in that case. So the way that samples are taken becomes very important. And so a bit of cleverness is required, I think, as well. So, for example, that includes knowledge about the organisms in question. Maybe they have certain habits that make it more likely to collect DNA in a certain area, say under a nest location potentially, compared to elsewhere, which might increase your chances of detecting it. So yeah, I think applying a bit of cleverness to it is required. Yes.
Is this research in many places now, or is this pretty novel for UVic?
So the question was, is this research just like for UVic, like from UVic, or is it more generally applied? The answer is it really is a worldwide phenomenon now. So environmental DNA is, so the first use of environmental DNA was back in 2008. And they were actually, it was in France, and they were looking at, so when I say first use of, it's in terms of using it for detecting animals. So they were actually looking at the invasive bullfrog in France and used this technique in order to identify where it was invading into. There are very active groups across Canada, as well as in the States and in Japan, in Europe, and in Australia and New Zealand. Yes, I mentioned Japan. Yes, so there are a lot of places that are starting to adopt it, yes.
Just the gentleman there.
Yes?
Curious, it just occurred to me, the temperature range before the DNA degrades. For example, I just thought about it coming from a hot spring, for example. How long does that remain viable before you get too many errors?
Yeah, so the question was about basically the effect of temperature on DNA, basically. So it turns out that actually temperature doesn't really, like temperature in and of itself doesn't really influence DNA too badly. Like DNA can kind of hang around happily. You can boil it, and you can freeze it. And gradually it'll degrade, but it takes a while for that to happen, just in those scenarios. But probably more of a bigger factor is things like damage by UV light. That can degrade it more quickly. And also scavengers, like bacteria, find, these are like hors d'oeuvres to, bacteria. They just love scavenging that stuff. Yeah, it's a great question. Yep.
How do you select for sharp-tailed snake or whatever? I mean, you've got hundreds of species on that swath.
Yeah.
How do you select for the sharp-tailed snake?
Okay, how do we select for a sharp-tailed snake? We've got hundreds of species. How in the world can we make a specific assay for that? Yeah, that's a great question. And what we do is we utilize the barcode of life. So we all have our own specific barcodes, but we as humans have common barcodes. In other words, when I say barcode, I mean DNA sequences in our cells. And those sequences differentiate us from a fish. But believe it or not, there are some sequences that are very much like a fish, so we want to avoid those ones because we don't want to accidentally identify those parts. So what we do is we take those DNA sequences and we line them up. And we choose areas that we feel, based on available evidence, that seem to be unique to the target species, in this case sharp-tailed snake, and that do not appear in human, which is a common contaminant. Who would have thought, right? Same with dog or cat, but also other snake species. And you can imagine that kind of approach is only as good as our knowledge is of the sequences that are available to differentiate between them. And that is a sort of hit and miss right now for databases for this kind of thing. And that's one thing that the iTrack DNA project is going to address, for Canadian species anyways, to augment our library of DNA sequences representing different species, so that we can actually develop better, more reliable tools. Yep. Yeah, so the question was, is there an international database so that people can access it, like publicly accessible databases, so people can actually use that information for whatever they need, right? There are databases available. They're limited by how much information is in them. One that's very prominent is the BOLD database, the Barcode of Life database, that's out of Guelph. So Canadian, woohoo! There's the National Center for Biotechnology Information, NCBI, which is a branch of the National Library of Medicine in the States, which is a big repository. It's publicly accessible. Those are two prominent examples. And really the drive is to make these sequences publicly accessible so people can use them for many things.
Just the lady there in the scarf.
Oh, yes. Hello.
Hi. Has our time in the past 2 years, this COVID time, taught you something that you're applying now in what your own research is right now?
So, could you expand on that question? I'm not sure.
Well, there's been so much international collaboration. There's been change that's happened in the research world so fast that seems to may apply to the kind of research you're doing right now. Like, is there lessons that have happened over the past 2 years that you feel like you can apply to the research that you're doing in the future?
So the question was are there lessons that we have learned, like through our COVID experience, that could be applied to the research that I'm talking about? I would say absolutely. One is simply that we can communicate very effectively through Zoom. That's a new twist, yeah. But the other thing that's sort of a corollary to that is that I sit way too darn long because like there's like one Zoom meeting after the other. So I mean that's it's sort of a facetious kind of response, but in actual fact, communication between scientists, between team members, and collaborators has really been augmented by that experience. Like we know that we can interact in a more real time without having to travel far distances to be able to interact. But having said that, having that face-to-face interaction, you can't replace it. And so I've learned that to miss that, and that's really important. From the technology side of things, I think it's this eDNA approach has been strengthened hugely because like using qPCR, for example, as a highly sensitive method for detection for environmental DNA, which literally, like I say to my students, like what we're trying to detect is like a 15th amount of DNA in our sample, and they're like, well, what's a 15th? Well, it's about that much. But like really tiny amounts, literally one molecule of DNA in a sample can be detected by this method. It's that sensitive. But you have to have all the checks and balances in place to make sure that when you detect something, it's actually real. So you have to have controls and that, very important to do so. And I think the COVID experience has just augmented that reality because many of the tests are based on the same technology.
Let's maybe have just one last question. There's a gentleman there at the back.
Hi there.
So this might be outside of the scope of your expertise, but this is the number one question that I have regarding DNA. What is the mainstream sort of opinion in academia regarding designer babies?
What is the mainstream opinion about designer babies? Wow, okay. In academia.
There's a lot of discussion going on the internet, but I want to know, like in academia, how do people approach this problem?
Well, creating designer babies is illegal.
I've actually got an answer to that.
Sure.
Do you want me to pitch in?
Yeah, for sure.
I mean,
yeah.
it's not quite to do with the topic of the presentation, but I don't want to dismiss it. You know, scientists provide science, right? Humans come to decisions as a corporate entity, right? So when Karen mentions it's a legal question, it's an ethical question, and we debate those. We can come up with consequences as scientists. We can present, this is the consequence of making this decision based upon our best evidence. And then our agencies, one of their functions is to help us decide, right, what is right and what is not right? What are we going to pursue? What are we not going to pursue? So I think it's important to answer that question. You know, we are not, as a scientist, one is not the authority here. One can provide input to the discussion, but you know, we as a community have to decide, right? Just like we do over, you know, COVID-19. Right? How do we approach that? Scientists have provided science, but we as a society have to reach the answers together, right? All right. Can I just put in one more question? I've been dying to ask this, and it's a crazy one. This is presenter's privilege, I'm sorry, right? So you filter water for DNA. Has anybody been crazy enough to filter air?
Absolutely, yes.
Okay, and what
it
did they find?
It works.
Oh, interesting. Okay, thank you
Yes.
for answering my question. I will not pursue it any further. Before we thank Karen one last time for a wonderful presentation, let me just remind you, we are back next week. Okay, we have another presentation. It will be on the Silent Genomes Project, which is more to do with the interpretation of DNA and healthcare for humans. Okay, so it's DNA month, but it's a very different kind of approach in that sense. So please join us next week if you're available to. It's all up on Eventbrite. You must have all got the emails as well. I hope to see you all again next week, but until then, please join me in thanking Karen. Thank you very much.
Thank you.